Hi everyone, so today we're going to head into lab number 3 and we'll focus on X-linked and 3-point crosses. Now I want you to know that these are some of the topics that students sometimes get a little scared of, especially the 3-point cross, because it can look kind of complicated and scary when you see it written out as a question on a test or in a textbook. But we're going to go through some steps that make it a little bit easier. But the first thing we're going to focus on are the X-linked crosses. Now, in order to understand X-linkage or X-linked crosses, first I wanted you to visualize the difference between the traditionally male and female sex chromosomes. Okay, so there are two sex chromosomes. There is the X chromosome and the Y chromosome. Okay, so XX will be the genotype in terms of those sex chromosomes that we associate with the female organism, and XY are the chromosomes that we associate with the traditionally male characteristics of an organism. Okay, and now we're not going to go into gender identification. That is something separate. Right now we're simply focusing on the chromosomes leading to certain characteristics. Now I want you to take a look at the X and the Y chromosome because you'll notice major difference in terms of how these two sex chromosomes look. You notice the X chromosomes have extra series of alleles or of genes not found on the Y chromosome. It's a much smaller, lesser looking chromosome. Now keep in mind that then affects organisms when we map out inheritance, because you'll notice the difference in these two possible genotypes. You know, the organisms that get 2 copies of an X and no Y, and the organisms that get 1 copy of an X and 1 copy of a Y. So when we look at an X-linked cross, first of all, what it means is that we are going to be studying a trait for which the genes are found on the X chromosomes. Okay, and so that means if you have a mutant allele, it will be found on an X chromosome. Now that means that you need to think about which chromosome each gender is getting from their parents. So is it an organism that's going to have 2 X chromosomes as its gender, or is it going to have an X and a Y? And since this is a gene only found on the X chromosome and not on the Y chromosome, that can greatly affect what we see. Now, when we do this cross, we can map it out with Punnett squares, and when we do the X linkage cross, we do this in a way where we set up 2 different crosses, because like we said, where the mutation is matters. You know, whether it's an organism that has XX or XY matters. So we're going to first draw out the first cross that we would do for an X linkage cross. Okay, so the first cross, we would have our P1, okay, that's the parental generation. And we would have in class cross wild type male with a white-eyed female. Okay, so that's the male symbol versus the female symbol. Now, when we're writing out their genotypes, because it's X-linked cross, we first have to write the sex chromosomes and then fill in whether it's wild type or white-eyed. So for the male fly, the way we would first write out the sex chromosomes is XY. And then for the female fly, we would write out XX. Now we're talking about eye color that is X-linked, so we will put the allele for that trait on only X chromosomes. The Y chromosomes will not get one of those alleles. So the first fly we said is a wild type for this X-linked trait. So that fly's X will have a wild type allele, which remember for fruit flies we're going to show as a plus symbol. The female is white-eyed. Now that X-linked mutation is also recessive, which means if she is looking white-eyed, then she has a mutant white eye allele on both of her X chromosomes. Okay, so we would cross the wild-type male with the white-eyed female. And then you can do a Punnett square to see what the offspring from that cross would be. So you do your Punnett squares just like you normally would, where you put the gametes from one parent on one side and the gametes from the other parent on the other side. So our first parent, we said, has an X plus and a Y chromosome. The second parent has XW and XW. Okay, and then you would fill this out just like you would any other cross. Sorry, I should have made it a little bit bigger for you to see. Okay, and we have XW with a Y, XW, X plus, and XW with a Y. Now, I'm assuming you've had some practice with the Punnett squares in lecture, and we did some in lab last time. So I filled that in, assuming that you know how to do that. If you are uncertain, please send me a message and remind, and I will go over how we got the centers of those Punnett squares filled in. And what you are noticing in this result, in the results within this Punnett square, is that all of the females, so this half, those are the females because they're XX, notice each one has at least one plus mark, one wild type allele. So that means that all of the F1 females, what do they look like? They look like they're wild type. Whereas all of the F1, meaning the offspring of the parents, of the males, all those F1 males, what do they look like? They have no plus mark anywhere, which means all F1 males are white-eyed. Okay, they're all mutants. So now these would be our F1 to then cross together, because again, I know flies are gross. They don't realize that a lot of them are their own siblings in the F1. So we're just going to cross those F1s together. Okay, so we're going to write out our F1 over here. So like we just said, all of those males were white-eyed. Okay, white-eyed males. And we knew that because they were XW with the Y. And then we cross them together with all of those wild-type females, which you saw were heterozygous. They had the XW and the X plus allele. Okay? So now what you would then do is make a Punnett square of those F1 crosses. Okay? And I'm going to do a zigzag line so you don't get confused with the previous cross. So our F1, the way that we would set up this Punnett square to see what the F2 will look like is just like always. We put one parent on the top portion, so our males are XW with a Y chromosome, and the female F1 parents are XW with an X plus. So those are their gametes that they can make, and then we just fill it in the center as we always would, matching up which alleles go into that square. Okay, plus XW, X plus from one parent, and a Y chromosome from the other parent. Okay, so we filled in all of these. Now you notice you have your genotypes, and then we can think about the phenotypes from this cross. Okay, so when you look at this There are a few different ways that we can ask about those F2. Now keep in mind the F2 are all of the flies that are in the center of this Punnett square, those 4 options. So I could either ask you about the percentage of males or the percentage of females expected to look a certain way, or I could ask you the percentage of all of the offspring to look a certain way. Okay, so first we're going to go based on percentage of each of the different flies that we're looking at. So when you look at the male offspring, so we're looking at this side, you'll notice that the F2, what do they have? 50% of the males. So 50% of the F2 males. Okay, so we're only looking at the males right now. So half of those males, what do they look like? Half of them are white-eyed, which is here. Half of the males are wild type. We're ignoring the other half of the Punnett square because we're only looking at the males right now. So 50% of the F2 males are white-eyed, and 50% of the F2 males are wild type, meaning they have red eyes, okay? And we can also do the same thing for the females. You'll notice that 50% of the females, what do they look like? White-eyed, okay, because we're only looking at the first half of the Punnett square now. Okay, so 50% of those females are white-eyed, and 50% of the F2 females are wild type, again meaning red eyes. Now, like I said, I can ask that question different ways. So if instead I asked what percent of F2 offspring, meaning now you're looking at all 4 of those squares in the Punnett square, what percent of F2 offspring is expected to be male with white eyes? What would you say? Well, you'd look at all 4 of those squares, and how many out of 4 are males with white eyes? Only one of those squares, right? Right over there. Only one of those squares is male with white eyes. So 1 out of 4, which is 25%. Okay? Or we can say 1 out of 4. Either one. Okay? So on a quiz, I might do it as the fraction just so that you look at your Punnett square and you say, okay, 1 out of the 4, or 2 out of the 4, 3 out of the 4, rather than having to convert to any percentage. Okay? So this is our first possible cross in which the mutation started out in the P1 generation on the female. Okay? If you have any questions about how we tackle any of these problems, so I can give you a cross. I can say you are crossing a white-eyed male with a heterozygous female, and you should be able to answer questions about the F2 like we just did. Okay, if you have difficulty with that, if you're not able to answer those kinds of questions, then just send me a message and remind, and we'll go over this again. Now, I could give you any kind of cross, okay? I can say a wild type male crossed with a white-eyed female. I can say a white-eyed male crossed with a white-eyed female. You know, all these different crosses you should be able to make Punnett squares for, okay? So we're going to move on to the next one, the other option for our P1, and see how swapping the P1 gender can affect our final results. Okay, so now, as I mentioned a few seconds ago, because this is an X-linked cross that we're looking at, the genders of the P1 can greatly affect the final outcome of our F2 and our different percentages. So we're going to visualize how changing the P1 now suddenly can change what we saw before. So now, instead of having the male be wild type, our P1 is going to have the mutation be in our initial male. So we start out with a white-eyed male now, crossed with a wild-type female, okay? And notice when we say wild-type, we have to specify that that is homozygous, okay? Because females have 2 X chromosomes. So if I just say wild-type, you don't know whether she's homozygous or heterozygous, meaning she's carrying a mutation, which she is not because she's homozygous wild type. So the way we draw this out for their genotypes, males, again, we start by writing XY, and since he is showing white eyes, that means that he has no plus alleles present. So he's just got the white-eyed mutation on that X chromosome. You then cross him with the wild-type female, and we said she is homozygous, meaning she's got wild-type plus mark on both of her X chromosomes. Now, just as we did before, we're able to set up a Punnett square for this cross. Okay, and again, you put one parent on top, and then the possible gametes for the other parent along the left-hand side. Okay. So now, when you fill in each of the squares, again, I'm not going to explain this because I'm assuming you are now already pros, but if you're uncertain, just send me a message and remind, and I'll help you fill in the squares. Okay? I always feel like I'm Bob Ross when I'm narrating as I'm drawing out the Punnett squares. Okay, so no happy little trees. Instead, we have happy little sex chromosomes. Now, what you'll notice is we're already starting to see differences from the first cross that we did on the previous slide. In the previous slide, when the female was our mutant in the P1, you notice we saw an F1 generation that had a variety of phenotypes. There were some wild-type offspring and some white-eyed. But now, what do you notice? All 4 of these squares inside there representing the F1, all of those offspring now look wild type. Each of those 4 squares has at least 1 plus mark, meaning all 4 look wild type. So we'll just make note of that, that all F1 offspring look wild type, meaning they have red eyes. Okay, so when we do quizzes and stuff like that, I could ask you, what percentage of the offspring or what fraction of the offspring would, look a certain way. And in this case, if I said you cross a white-eyed male with a homozygous wild-type female, you should be able to do that Punnett square and realize all F1 offspring 100% look wild type. Okay? Now, we then cross those wild type F1 together to make our F1 cross. So we have the wild type males from that cross, and we're crossing them now with heterozygous wild type females. So heterozygous wild type females, because notice each of these females in the F1 carry 1 allele of each type, 1 wild type, 1 white-eyed. Okay, so we draw out our genotypes. Wild type male only has 1 X chromosome and it is wild type, and then he's got a Y chromosome, and we cross him with heterozygous female. So she's got 2 X chromosomes, but when you see heterozygous, that means she's got 2 different alleles on each chromosome. So she's got a plus mark and a white-eyed mark. So now with that cross, we can draw a Punnett square to try and visualize the F2. So again, I'm going to zigzag separate our previous cross so we can look at making the F2 now. Okay, so we're going to do our Punnett square to see what F2 we have possible. Again, we draw genotype of one parent, so I always draw the male up here, and we have our heterozygous female on the side here. Okay, and then you just fill in as we keep doing. Okay, x plus, x plus matched with a y, xw matched with an x plus, and xw matched with an xy. And now what do you notice? Well, let's look at the males first like we did last time. So our F2, what do you notice about the males? Well, 50% of the males, so put 50% of males, they look wild type, right? Because these 2 squares are our males. 1 out of 2 of those squares has a plus mark. The other 1 out of 2 has only a W. So 50% of males look wild type. 50% of males look white-eyed. Now, what do you notice about the females? Okay, so now we're just looking at the female half. Well, both of those squares have at least 1 plus mark. Okay, and as soon as you see a plus mark, that means they look wild type. So now 100% of the F2 females look wild type now. Okay, so that's different from the previous results. Okay, there are no white-eyed F2 females possible in this cross, theoretically. Okay, so usually in class when we do that cross, if any students end up with a white-eyed female at the end, we realize that they had some cross-contamination somewhere along the way where they miscalculated a gender when they were putting the flies into those tubes. Okay, now again, I could ask the questions in different ways, so we can look at all 4 of the squares in this F2 set of possible offspring, and instead of saying what percentage of the males are wild type or what percentage of the females are white-eyed, I can say what percentage of the F2 is white-eyed. Okay, so can ask different ways. So for instance, what percentage of F2 offspring in this cross, and I'd always tell you what flies you're crossing together. So what percentage of F2 offspring when crossing a wild type male with a heterozygous wild type female, what percentage of F2 offspring would have white eyes? And then instead of just looking at one gender, you look at all 4 of those squares, and what do you notice? Only 1 out of the 4 squares has white eyes. So I would allow you to write 1 out of 4 instead of a true percentage, or you can say 25% of F2. And again, if I was writing this out on a quiz, I would first, at the beginning of that question, write out the exact flies that you're crossing together. I'm not doing that right now because it would look redundant on this slide, but keep in mind in your notes, if you want to write out the whole question, in front of what percentage, throw in if you cross a wild type male with a heterozygous wild type female, so heterozygous for white eyes, what percentage of the blah, 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 blah. Okay? Now, notice that in this cross, when you're doing X-linked crosses, the mother to son inheritance becomes a big deal because the sons, they have to get their Y chromosome from dad. They only get their one X chromosome from their mom, okay? And that means that they only need one mutant allele in order to show a mutation that's X-linked. So sons have it bad in that sense, okay? In fact, what you notice is we refer to the female in this cross as heterozygous, but you've never heard me in any of the X-linked crosses refer to a male as heterozygous because he's only got one X chromosome. So he can't be heterozygous in his genotype because that would require two different alleles. He's only got one allele. So make a little note somewhere. I'm going to do it in this big empty space. Males in X-linked crosses cannot be heterozygous. They can only be called hemizygous for a mutation if it's X-linked, okay? So keep that in mind. If you ever hear hemizygous, they're referring to a male who has a mutation in the X chromosome. Okay, so again, if you have any questions on how we did this or you want to practice the questions the way I was asking it at the end of the problem here, just send me a message and remind, and I'm more than happy to help. This here is just a visual textbook kind of example of the crosses we were just going over, but notice their cross A and B is swapped from ours. So what you can do is cross out cross A and write that that was our second cross that I showed you, whereas cross B was the first cross that I showed you. And then when you look at this, you kind of get an example of what the flies look like. And you'll notice that depending on changing the P1, you know, all you did was swap out which parent had the mutation, it had a huge impact on the final results that you saw, especially in the fact that that second cross we went over, which is their cross A, you notice you completely got rid of one of the options. There was no longer an option for white-eyed females. Okay, so gender plays a big role in terms of these X-linked crosses and in terms of inheritance. You know, are you inheriting an X-linked mutation from your mother or is it coming from your father? That can have a drastic effect. Okay, again, any questions, just send them my way in the Remind app. Now, I just wanted to give you two examples. There are multiple examples, but I wanted to give you two of the most common examples that we mention when we talk about humans having X-linked disorders, where, you know, the gender of the parent, the gender of the offspring, in terms of this inheritance makes a big difference. The two big examples we do are red-green color blindness and hemophilia. And I always feel bad when I do this slide because there have been times when I've taught the lecture where suddenly a student realizes that they have red-green color blindness and they never noticed it before. So the way to check whether or not you have red-green color blindness is you take a look at the figure over here, and if you can see that there's a number in the middle of it, then you're all good. If you cannot see the number in the middle, which my brother has that problem, that means that you may have inherited red-green color blindness. The other example on this slide is hemophilia. That is a clotting disorder. Okay, so basically the patient is not able to properly clot their blood because they're missing one of the important clotting factors. Okay, we're not going to go into the details of each of these disorders right now. I just want you to know them as examples. So if I asked you which of the following is an X-linked disorder, and I gave you some examples, be able to identify either red-green color blindness or hemophilia. So the last point that I want to make regarding sex-linked or sex-based inheritance is that there are a few different examples. The first one is sex-limited inheritance. So that's basically where you only see a particular phenotype in one of the genders. And the big example that they use for this is the feathering of some birds. So for instance, when we look at this picture over here, right away you can tell which of these two birds is the male bird, and that's this guy over here. You can tell based on the very ornate, big, bright, colorful, extra feathers that they will have in their pattern. And the reason that is that the male has to be able to attract the female in order to try and pass on its legacy, whereas the females get to have the luxury of being a bit more selective in choosing their mate, because the females, if you think about it, reproduction has a much greater commitment and cost to the female than the male, because that female now, once she is pregnant, she has to carry that offspring for many months depending on the organism. And not only does that put a physical toll and danger on her body, but in the evolutionary sense, out in the wild, it also makes her more vulnerable. So if she's going to go through all of that cost, that, the heavy costs that are associated with pregnancy, she wants to make sure that her and her offspring have the best chance. So she will try to pick the most evolutionarily fit male. And one of the things that she'll look for is the most ornate, big, beautiful looking feathers in the world of the birds. Okay, males, on the other hand, they get to just spread their seed wherever they want, trying to maximize their chances of passing down their genetics. But they don't have the cost that the female does. Okay, even if you think of humans, the human female, she's pregnant for 9 months. And not only does that put a great toll on her body, but, again, it makes her more vulnerable to any dangers around her. Okay, whereas the male does not have that during that time. Okay, so feathering is sex limited. For the birds, usually it will just be in the male that you see those special ornate feathers. There's also sex-influenced inheritance, where basically either gender can demonstrate a particular phenotype, and instead of it being based on the sex chromosomes themselves, it's actually influenced by hormones. So for instance, differences in testosterone versus estrogen. And the big example that we usually use is male pattern balding. In a lot of textbooks, they like to use the picture of Jean-Luc Picard in Star Trek. I like to use family pictures because, as you can see, here we have 3 generations of Esposito males. And my brother here demonstrates a great example of male pattern baldness. Okay, and you can see it great in this bottom picture here, because if you cut this picture in half, you have 2 Esposito males on one side, and you have 2 Esposito females on the other side. And what do you notice? Look at that male pattern balding right here. And yes, I will have to show them this slide at some point. Whereas the females, look here, Nana is almost 90 in this picture, and look, that is her real head of hair. Nice, thick, full, completely filled in. Whereas the males, unfortunately, do not have that. And so certain traits, certain phenotypes, they're greatly affected based on things like hormone and your sex chromosomes. Okay. Okay, that was a lot of information, and you are about to get hit with a whole lot more of information when it comes to the 3-point cross. So I split this lesson into 2 videos. You can take a quick break now and then head over to the second half of the lesson.